Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the next lecture in the third unit of Plant Biotechnology course. In this unit, we are learning about the plant protoplasts, how these plant protoplasts are being used to study the basic biological phenomena in plant sciences, and there are several commercial applications for crop improvement or generating new crop cultivars. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University. In JNU, I teach plant biotechnology and genetic engineering at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. And this project has been sponsored by DTH Swayamprabha, MHRD, New Delhi. So in this lecture, I will talk about the protoplast fusion for generation of somatic hybrids, various methods of protoplast fusion and their advantages or various limitations. Then we will talk about the different types of hybrids like asymmetric and symmetric hybrids. And finally, towards the end of the lecture, I will briefly discuss about various segregation patterns of cytoplasmic genomes after the somatic hybridization. First, a quick recap of the protoplast preparations that we discussed in the last lecture. A plant cell, when stripped of its cell wall, is called a protoplast. These are also called as naked plant cells, referring to all the components of the plant cells except the cell walls. Depending upon the method used to strip off the cell wall to extract the protoplasts from these cells, these can be grouped into two categories. In the mechanical methods, where we plasmolyze the tissues and cut it with the sharp blades to release the protoplasts. However, the enzymatic methods are more popular as it is easy to execute and yields are higher. Basically, we incubate the tissue with the cocktail of pectinases and cellulases, that is the cell wall digesting enzymes that digest the cell walls and release the protoplasts. Then we also talked about how to remove the debris and broken protoplasts from this protoplast and tissue mixture and purify the protoplasts, followed by testing their viability using various methods. We discussed about how to culture the protoplast and various factors that significantly impact the protoplast culture, like the health of the protoplasts or health of the source tissue from where these were extracted, composition of the in vitro culture media. There are few specific requirements for culturing the protoplast, and finally the maintenance of the osmoticum in the culture media, which is also very important for maintaining the protoplast. At the end of the last lecture, we talked about various applications of protoplasts in the basic biology and for the commercial purposes too. Today we will talk about the somatic hybridization and application of the protoplasts that has been used to study the basic biological behavior, various basic biological processes, as well as has been used commercially for the crop improvement, and several new cultivars have been regenerated through the somatic hybridization. Somatic hybridization. The process of the fusion of two protoplasts that are isolated from the somatic cells of two different plant species or cultivars to regenerate the hybrid plants is called as somatic hybridization. As it involves the protoplasts from two different plant species or cultivars, first step is to select the relevant plants. So usually there must be some advantage from this fusion, like if these plants may have some specific characters that we want to bring together in the hybrids, or interest may be to study the basic biological phenomena, how the different components of these two different somatic cells they interact to each other. Next step is the selection of suitable explant or suitable tissue from where to prepare the protoplasts. As all the plant cells are totipotent, theoretically protoplasts can be prepared from any of the plant cells. However, actively growing young tissues are the best for this purpose. Actively growing cells in the suspension cultures and leaf mesophyll cells have been found most suitable for the preparation of protoplasts and are commonly being used for this purpose. So here is the leaf tissue which is selected for the purpose, that is protoplast preparation. Small segments of the leaves are prepared, and these segments then are incubated with a cocktail of cell wall digesting enzymes. Usually cellulases and pectinases are successfully used for the cell wall digestions in most of the plant species. Enzyme digests the cell wall and protoplasts are purified. Now these purified protoplasts from two plant species, they are mixed together and fusion of the protoplast is induced. Several methods are used to induce the protoplast fusion, and we will learn about these methods shortly later in this lecture. The fusion of two protoplasts results in a hybrid cell which are cultured to regenerate the hybrid plants. So in this way, the protoplast fusion can be used to mix the genomes from the somatic or the vegetative cells without the process of sexual reproduction, and the hybrid plants can be regenerated in vitro cultures. In order to induce the fusion of the two protoplasts, several methods are being used, and depending upon the type of the force involved or what type of the inducer is used, these fall under following three categories, that is spontaneous fusion, mechanical fusion and induced fusion. Depending upon the type of inducers, these are of two types, that is the chemofusion where the fusion is induced using the chemicals, and electrofusion, that is when the fusion is induced using the electric field pulses or electric current. The first method is the spontaneous fusion. During the protoplast preparation, the protoplasts by chance come close to each other. Plasma membranes are fluid and simply the physical contact among the adjoining protoplasts may be sufficient to fuse the plasma membrane together, making a hybrid cell. Plant cells are connected to each other by the plasmodesmata. Now, plasmodesmata are the small channels that connect the cytoplasm of the adjacent plant cells. It means that the plant cells are not completely isolated by the cell walls, but the living bridges between the cells exist in the form of plasmodesmata. These bridges run through the cell wall and the middle lamella and allow certain molecules to pass directly from one cell to another cell, and that may be important for the cellular communication. During the enzymatic digestion of the cell wall, the plasmodesmata enlarges because of the removal of cell walls. This enlargement of the plasmodesmata may allow the movement of larger organelle to the neighboring cells, and as the cell wall digestion proceeds, even the complete fusion of the two adjacent protoplasts can take place, resulting in a hybrid fusion body. Usually these type of fusions occur when the protoplasts are prepared and therefore the protoplasts of the same species are involved in these type of fusions, and the fusion products are called as the homokaryocytes or the homokaryones, as the hybrid bodies or the fusion bodies contain the same type of multiple nuclei. That is, origin of these nuclei is from the same parent. More than two cells may fuse together to form the homokaryocytes that have more than two nuclei. In exceptional cases, these homokaryocytes may contain up to 30 or 40 nuclei too. That means usually large number of protoplasts may fuse together during the enzymatic digestion of the protoplast preparation, and hence it is very important to prepare the protoplasts or purify the protoplast carefully. Further, it has also been observed that the frequency of the spontaneous fusion of the protoplast that result in multinucleated fusion products, it may vary with the type of source tissue used for this purpose. Higher frequency of multinucleated protoplasts has also been observed when the protoplasts are prepared from the suspension cultured cells. Next is the mechanical fusion. That means external mechanical force is used to bring the protoplast together for fusion. Isolated protoplasts usually have an overall negative charge on their surface, and hence they repel each other, that prevent their fusion. Therefore, in order to fuse these protoplasts, these need to be pushed to come closer so that their plasma membrane can be in contact with each other. Therefore, micromanipulator and micropipettes can be used to bring the isolated protoplasts together in intimate physical contact, and these procedures are performed by looking under the microscope. Or simply a gentle tapping of the protoplast may result in the protoplast fusion. Like protoplast suspension may be put in a small depression on a slide, and the gentle tapping of the slide can provide the required push for the protoplast fusion, or even these may be performed in a small tube. However, the frequency of the fusion is very low, and further, as these are the crude procedures, the reproducibility of the protoplast fusion is also very low and can vary significantly with the tissue types too. The protoplasts of the Acetabularia were fused mechanically and hybrids were generated successfully. Acetabularia is a type of green algae. These are single-celled green organisms. However, they are gigantic in size, and therefore the protoplasts are also bigger. Many authors consider and discuss the mechanical fusions under the induced fusion, as the fusion is induced by the use of external mechanical force, whereas some other authors discuss it as a separate category. Next is the induced fusion. Induced means that the external stimulus is used to achieve the fusion of the protoplast, and depending upon what kind of stimulus is used, it can be categorized into two types. One is the chemical fusion or chemofusion, where the chemicals are used to achieve the protoplast fusion. And second is the electrofusion, where the electric pulse or electric field is used to achieve the protoplast fusion. So the first one is the chemical induced fusion or chemofusion. Now, because of the overall negative charge on the surface of the isolated protoplast, they have a strong tendency to repel each other. And so if we want to bring these protoplasts in the close vicinity of each other so that their plasma membrane can be in contact and fuse, we need to reduce or negate the negative charge on the protoplasts. Several different chemicals have been used successfully for the protoplast fusion. Some of the commonly used chemicals for this purpose are polyethylene glycol, calcium with a higher pH, sodium nitrate, dextran sulfate, or polyvinyl alcohol. Even certain authors have tried use of lysozyme and fatty acids successfully for the protoplast fusion. However, these are not very commonly used. The chemicals which are used for the purpose of fusing the protoplasts are called as the fusogens, and they may act either by reducing the negative charges on the surface of protoplasts, or their presence may result in the electrostatic forces that attract the protoplast together and result in agglutination of the protoplast. The use of these fusion inducing chemicals causes the isolated protoplast to adhere together, resulting in the agglutination and finally the fusion of the protoplasts. So the first one is the polyethylene glycol induced fusion. It is also called as PEG. PEG is one of the most widely used fusion inducing chemical for the somatic hybridization. Polyethylene glycol is a non-ionic surfactant whose molecules are highly water soluble. It has slightly negative polarity and forms hydrogen bonds with the positively charged groups on the protoplast surface like polarized groups of proteins, carbohydrates, or water. It is a high molecular weight compound and its molecular weight ranges from about 1500 to 6000. Now, polyethylene glycol molecules can act as a molecular bridge between the adjacent protoplasts. Though PEG has a negative polarity, it interacts with the positively charged groups of the membrane and can hold the protoplasts together or cement them together. Here are two protoplasts that have been extracted from the leaves of two different plant species. Addition of the polyethylene glycol causes the agglutination of the protoplasts, that is, protoplasts come in contact with each other or it holds the protoplasts together. The concentration of the PEG may vary from somewhere 15 to 45%. However, around 25 to 35% concentrations of PEG usually work successfully for most of the plant species. Later, when the polyethylene glycol molecules are eluted or diluted out, it results in the fusion of the two membranes at the point of contact. Fusion body makes a dumbbell-shaped structure and soon becomes spherical. At this stage, when the two protoplasts fuse and the fusion body has two nuclei from two different parents, these are called as the heterokaryons. As the PEG is removed or eluted completely from the solution mixture, the fusion bodies are deplasmolized and cytoplasm of the two cells mix together to form the hybrid cell. It results in high frequency heterokaryon formation and has high reproducibility. Therefore, it is commonly used for protoplast fusion and is one of the most popular fusion used for somatic hybridization. Next is the use of the calcium with higher pH or the alkaline conditions for the fusion of the protoplast. Now, this method was originally developed by Kahler and Melcher in 1973 for the tobacco protoplasts. The protoplasts from both the parents were mixed in a liquid medium having high concentration of calcium ions and alkaline pH. pH is usually in the range of 8 to 10.5, and then these are incubated at higher temperature around 37 degrees centigrade for about 40 to 50 minutes. High concentration of calcium ions with alkaline conditions and incubation at high temperature induces the fusion of the protoplast. Calcium ions as well as the higher pH both neutralizes the surface charge on the protoplast. Calcium interact with the negatively charged groups on the plasma membrane of the protoplast. These charge neutralized protoplasts no more repel each other and they can adhere to each other. That means the protoplasts can come closer to each other, leading to agglutination. This results in the membrane fusion of the protoplasts, which results in the protoplast fusion of forming the heterokaryons or homokaryons. Further, the calcium with higher pH and PEG have been used together for the protoplast fusion and has been found to enhance the efficiency of protoplast fusion significantly, and now is commonly used for protoplast fusion in many plant species. It has been suggested that the calcium can bind the polyethylene glycol molecules in the solution mixture, and it also interact with the protoplast surface at the same time. So basically the calciums form a bridge connection between the PEG molecules and protoplasts, and therefore it holds the protoplasts together and enhances their agglutination. Let's have a look on the mechanistic details of the chemical-induced protoplast fusion. Here are the two protoplasts that have been prepared from the two different parents, that is species A and species B. Protoplasts have been marked with the different colors for the identification purposes. Equal number of protoplasts from both the plant species is mixed together. When PEG are applied in the solution mixture of the protoplast, if the plan is to use the PEG and calcium together, required amount of calcium salt is also added. Usually calcium chloride is used for this purpose. It brings the protoplasts close to each other and hold them together. Various explanations have been suggested to explain the mechanism of protoplast fusion. The plasma membrane is made up of lipids and proteins, and the use of external fusion or fusion inducing chemicals causes the configuration changes in the membrane proteins and lipid. This increases the overall fluidity of the membrane, and when the two protoplasts adhere to each other, intermixing of the lipid molecule can occur at the point of contact of two protoplasts, and the membrane can just fuse from the adjacent protoplasts, leading to the fusion bodies. Like here, when the PEG and calcium are used, calcium ion can interact with the negative groups on the membrane and reduces the negative charge on the protoplast surface. In addition, it can interact with the PEG that has negative polarity too and act as a molecular bridge between the polyethylene glycol and the protoplast. This not only helps to reduce the charge on the protoplast surface, however, it also helps to hold the protoplast close to each other. When the PEG is diluted out or eluted, it results in the change in the surface potential and enhances the fusion of the adjacent membranes. Further, PEG has a strong affinity for water. Therefore, when PEG is diluted out, it may cause local dehydration in the area of the point of contact of two protoplasts, and this increases the fluidity of the membrane, which enhances the protoplast fusion. Protoplast fusion passes through a dumbbell shaped structure where the plasma membrane fuses at the point of contact. However, this is a transition stage and soon it changes to a spherical body. As the PEG is removed or eluted completely from the solution mixtures, the fusion bodies are deplasmolized and the cytoplasm of the two cells mix together, forming a hybrid cell. Finally, the hybrid plants are regenerated from the hybrid cultures in the in vitro conditions. However, agglutination is a random process, And it is not the only possibility that the two protoplasts that come in contact with each other would be from different parents. These protoplasts that agglutinate together may be from the same parents, like the two parents of species A may agglutinate together, or the protoplasts from the species V may agglutinate together, and these may fuse to make the fusion bodies in the similar fashion as discussed for the heterokaryons, or when the two protoplasts are fused from different parents. The spherical fusion bodies that are formed by the fusion of protoplasts from two different species, they contain the two nuclei which originate from the different parents, and these fusion bodies are called as the heterokaryons or heterokaryocytes. And the fusion bodies that are formed by the fusion of the same type of protoplasts are called as the homokaryons. Next is the electrofusion. The use of electric fields to achieve the protoplast fusion has become a competitive alternative to the chemical induced fusion. So in this case, a mild electric field pulse is used to induce the protoplast fusion. Protoplasts from both the parents are mixed in the equal ratios and are suspended in the liquid media having low conductivity. Usually calcium chloride with the mannitol is used for this purpose. Mannitol maintains the osmolarity of the suspension, and calcium chloride help in aligning the protoplast in the electric field. The protoplast density also play an important role, and about 500,000 protoplasts, that is 5 × 10⁵ protoplasts per ml, is an optimal concentration that can be used for most of the plant species. Low voltage current is applied in the range of 100 to 200 volts per centimeter. It induces or generates a mild dipole on the protoplast, and therefore the protoplasts align themselves in a chain parallel to the direction of electric field. And at this stage, the protoplasts or chain protoplasts, they appear like the pearl chains. In order to achieve the fusion of the protoplast, one or a few pulses of high voltage direct current in the range of 0.2 to 2 kV per centimeter are applied. It causes the breakdown of the plasma membrane of the two protoplasts at the point of contact and result in the fusion of the adjacent protoplasts. Electrofusion is very competitive to the chemical fusion or the chemofusion. It's rapid, more synchronous as compared to the mechanical methods or chemical induced fusion methods, and more importantly, it is very convenient to use. The chemical that are used as a fusions may have unintended consequences, like these may be toxic to the living protoplasts and can compromise the protoplast health. However, electrofusion is relatively non-toxic. The overall fusion efficiency is several fold higher than the mechanical or the chemical induced fusion methods. Further, its reproducibility is also very high across the independent experiments. How many protoplasts will align together to form a chain would depend upon the two factors. One is the distance between the electrodes, and second is the density of the protoplast in the solution. Number of the protoplast aligning in the pearl chains will increase with the increasing distance between the electrodes or increasing protoplast density in the mixtures. Therefore, these factors are critical for achieving the desired hybridization results. Various parameters of electrofusions needs to be optimized in a species-specific manner. Like the osmolarity of the solution should be in the right proportion. Protoplasts from different species may respond differently to the AC voltage for aligning the protoplast or the DC voltage for causing the fusion. Therefore, the protoplasts for different species may have the different requirements, and these needs to be optimized independently. Protoplasts used for the electrofusion should be of extremely good quality. Otherwise, these may burst open during the electrofusion, and bursting of the protoplast would release the cytoplasmic content or the salts in the solution, and it may affect the conductivity of the fusion mixture, thereby affecting the fusion efficiency too. In order to have only the heterokaryons, MicroElectroFusion technique has also been devised. So now the electrofusion is performed at the micro scale. Here the two protoplasts from the different parents that need to be fused are pre-selected and then are fused on a slide under a microscope. Selected protoplasts are transferred to the microdroplets of low ionic strength fusion medium. Micropipettes or micromanipulators are used to handle the droplets and mount these droplets on the slides. Electric field is applied through a micro setup of wired electrodes. And protoplasts again should be of extremely good quality, and independent parameters may need to be optimized in a species specific manner. So basically the mechanism of electrofusion or microelectrofusion remains the same, with only difference is that the microelectrofusion, everything is happening at a micro scale as we are fusing the two pre-selected protoplasts, and the procedures are performed under the microscope, and hence the micro setup is required. So the scale is the major difference between the two. However, the successful microelectrofusion, we will get the hybrids directly, which are heterokaryons or heterokaryocytes. No other procedure or the selection or the screening of the hybrids would be required, but at the same time it needs expensive setups too. Fusion of the protoplasts may result in two types of hybrids with respect to the nuclear genome. These are called as symmetric hybrids and asymmetric hybrids. So the first is the symmetric hybrids. If the regenerated hybrid plants contain the complete set of chromosomes from both the parents, these are called as the symmetric hybrids, and the process can be called as a symmetric hybridization. Symmetric hybridization had been the most common target of the somatic hybridization in the past, and a large number of symmetric hybrids have been produced in several plant species, like in many species of the citrus or the solanum, chrysanthemum or brassica, etc. Symmetric hybridization usually results in the production of polyploids, like this has been used to produce the several allotetraploids in citrus species. However, if the protoplasts are prepared from the haploid tissues, ploidy of the hybrids can be maintained as to that of their parents. Symmetric hybridization has contributed tremendously in the improvement of the citrus species. Even some seedless varieties have been produced by symmetric hybridization of haploid and diploid protoplasts. Symmetric hybrids between the distant species are usually undesirable, as the gene conflict may arise, and this causes the genetic imbalance in the hybrids. It interferes with the normal development of the hybrid plants, and such hybrids are usually unstable. Further, the symmetric hybrids carry large amount of unwanted genetic material. These hybrids have the complex genetic constitution and are required to backcross several times before establishing them as a new cultivar. Symmetric hybrids often show abnormal phenotypes, like these are usually recalcitrant to the regeneration in vitro cultures, and abnormal growth patterns are also very common. Even if the hybrid plants are regenerated, chances are that either these would be sterile or would have the low fertility. Shoot regenerated from the symmetric hybrid cultures often show recalcitrance towards the rooting, and therefore producing the plantlets on the in vitro cultures is often a challenge. Next is the asymmetric hybrids. Somatic hybrids have fewer genomic content from one of the parent and are called as asymmetric hybrids. So in the asymmetric hybrids, nuclear genome of one of the parent is eliminated partially. Such eliminations of the genomic fragments of the chromosomes occur spontaneously and are common in distant hybrids. The manipulations of the amount of DNA that would be eliminated, or which part of the genome or genomic DNA should be eliminated, cannot be controlled in these setups. However, elimination of the chromosome or the genomic content can be induced artificially before the hybridization, but again this elimination is uncontrolled, and we will learn more about this topic in the next lecture. Therefore, asymmetric hybrids where genome of the one parent is partially eliminated has a better chance to survive and has higher regeneration capacity too. Regenerated asymmetric hybrid plants are more stable and has a better fertility as compared to the symmetric hybrids. Usually, the donor genes that causes instability of the karyotypes are often eliminated in few divisions after hybridization. Therefore, both the symmetric and asymmetric hybrids have their own advantages and the disadvantages as well. Therefore, it would depend upon how distant are the two parents phylogenetically and what is the aim of somatic hybridization to choose whether to create the symmetric hybridization or the asymmetric hybrids. Next is the Cybrids. So the method of combining the nuclear genome of one parent with the cytoplasmic genome, that is the chloroplast genome or the mitochondrial genome of another parent, is called as cybridization, and the hybrid produced in this way are called as Cybrids. In the sexual hybridizations, cytoplasmic genomes are always contributed by the female parent, and there is no way to make the Cybrid-like combinations. When the protoplast fuse, the extranuclear genome of both the parents are combined in the hybrid cells. It is during the later stages of development when the cytoplasmic genome of the one parent is eliminated, or the chromosomal DNA may be destroyed in one of the parent protoplasts before the hybridization. In this way, these parent protoplasts will only contribute the cytoplasmic genomes to the hybrids. So in this way, we can also control which of the nuclear genome we want in the hybrids. Further, there may be interactions among the cytoplasmic genomes, and with the further development or the division of the hybrid cells, some of the cytoplasmic genomes are eliminated. Here are the two protoplasts from two different parent species. Again, different colors or the shapes are used to identify their origin. Finally, after their fusion, it may produce this kind of combination where the nuclear genome has been contributed by the one parent here in the species A, and the cytoplasmic genome are contributed by another parent, in this case by species B. Finally, the hybrid plants are regenerated from the hybrid cultures in the in vitro conditions. Next is the post-hybridization segregation events of the cytoplasmic genome. So when the protoplast fuse to form a hybrid cell, a set of three genomes from each parent interact in the heterokaryons or the fusion bodies. These are nuclear genome, plastid genome, and the mitochondrial genome. Post-hybridization, the interaction between the different types of genome may result in several different combinations of nuclear and cytoplasmic genomes in the hybrid plants. Usually, the nuclear genome of one parent is either partially eliminated or may be completely eliminated during the successive divisions before the regeneration of the hybrid plants, and therefore it results in the asymmetric hybrids or the Cybrids. However, heterokaryon may give rise to the hybrid plants with full copies of the nuclear genome from both the parents, and this result in the formation of symmetric hybrids. Now, in the cytoplasm of the heterokaryons, there are two types of plastid genomes and two types of mitochondrial genome too. With the further divisions, the cytoplasmic genomes segregate and may result in several combinations. Some of them are eliminated and some of them are retained finally in the hybrid plants. During the time when these multiple cytoplasmic genomes are together in the hybrid cells, there may be interaction between the genomes of the two parents before they segregate, and there may be an exchange of the genetic material between the genomes of two parents. That means the cytoplasmic genomes show interparental recombinations. Now, the exchange of this genetic material or the interparental recombinations are more common in mitochondrial DNA from the different parents, and it can generate several different combinations. Slowly, one of the many recombined genomes or the combinations is retained in the regenerated plants and others are eliminated or lost during the development. Interparental recombinations between the plastid genome of two parents are not very common and are rarely observed. Overall, these interactions can result in several different combinations of cytoplasmic genomes in the hybrids. Let's explore the various major possibilities of the cytoplasmic genome in the regenerated plants. Here are the protoplasts from two parents, that is species 1 and species 2. For the sake of understanding, only the cytoplasmic and nuclear genomes have been labeled. Also, these are colored with different colors in order to identify their segregation patterns in the progeny cells of the progeny plants. After the fusion takes place, the heterokaryons would carry all the three genomes from both the parents. However, in the pictorial representation, we are considering this hybridization as a symmetric event, and the various combination of the nuclear genome or the combination of the nuclear genome with the cytoplasmic genome are not considered. During the further divisions, cytoplasmic genome will be segregated and can result in the several possibilities. Here are the major categories of the segregation patterns of the cytoplasmic genomes. First possibility is that the mitochondria and plastids of one of the parent may be retained in the regenerated hybrid plants. So figure A shows where the mitochondria and plastids of species 1 are retained, and figure B represents the hybrid where the mitochondrial and plastid genomes of the second parents are retained. Third option would be that the plastid genome of one parent and the mitochondrial genome of the second parent may be retained in the hybrid cells. Figure C shows the chloroplasts are inherited from the species 1, whereas in this hybrid the mitochondria that are retained are inherited from species 2. Similarly, it may be otherwise, like option D shows that in this hybrid chloroplasts are inherited from the species V, whereas the mitochondria that are retained in this hybrid are inherited from the species 1. Earlier we talked about that there may be a significant level of interparental recombinations between the cytoplasmic genomes. That is, there is a significant amount of genetic material exchange that can occur between the different genomes of or the two genomes of the different parents. However, such kind of genetic exchange or interparental recombinations are more common in the mitochondrial genome and are rarely observed in the chloroplast genome. Therefore, in the figure E, mitochondria have been shown with a separate color, a color different from the ones used to mark the mitochondria in the parents. It represents that it carries the genetic material from both the parents. Again, these recombined mitochondrial genes can be present with the chloroplast of either of the parent, like in the figure E, these are from chloroplasts are inherited from the parent 1 or the species 1, and in the figure F, it represents the hybrid where the chloroplasts are inherited from the second parent or the species B. So these are the major categories of the cytoplasmic genome segregation patterns. So in summary, today we learned about the somatic hybridization, then various methods that are used to achieve the fusion of the protoplasts. We also talked about their advantages and disadvantages. Then we learned about the different type of hybrids like symmetrics and asymmetric hybrids and their limitations. And finally, we talked about various segregation patterns of the cytoplasmic genome after the hybridization during the regeneration of hybrid plants in the in vitro cultures. In the next lecture, we will continue the discussion on the somatic hybridization, and we'll learn more about the methods to generate the asymmetric hybrids or the symmetric hybrids, and selection of the hybrids from the mixture of various hybrid forms, and then the characterization of the hybrid plants and their application for the crop improvement. Thank you.